Thank you very much, Tomasz. It was a pleasure to work with you and to put this symposium together. And I'm excited to share some of my work in the field of neuroscience and in particular hypothalamus biology. Today, that's the first time here in Slovenia that I share that, more or less. And so in my lab, okay, let me see. So so I wanted to start with stress that Tomasz already mentioned before, that is somehow important how we react to stress on one hand, and several mechanisms have evolved for animals to survive that, and hypothalamus is in the center, and we somehow also with this in mind, designed this symposium. But in my lab, this is also the center of our sort of work in a way. So we try to understand molecular mechanisms that protect germ cells against stress, in the testis in particular, in male germline. We are interested how these pathways are hijacked by cancer cells and how we can use the insights we learn for advancement of cancer therapy. And for today's talk in particular, it's important we want also study the mechanisms that are disrupted and evolved for stress adaptation in neurodevelopmental diseases like Prader-Willi, and the functional thread that connects all of these seemingly unconnected tissues together are these proteins, depicted as this funny guy here, but they are called melanoma antigen genes, or MAGEs. And several our insights suggested us that these proteins evolved to protect germ cells against stress, and are oftentimes hijacked by cancer cells. I'm showing you a summary of two stories we recently published. But today we will focus on this MAGE-L2 protein in this family of 40 proteins or genes that we have in our genomes. So this is what I'm showing you, phylogenetic tree of human and mouse MAGE genes, and they come in two major flavors. Type 1 are genes connected to cancer and normally restricted to expression in germ cells, but type 2 are highly enriched in the brain, and MAGE-L2 is one of them, and main hero of the story today. And this is a busy slide just to show you that these genes are expressed in different organs, and expression-wise come into different major patterns. On the left are these cancer testis antigens expressed in testis, and on the right are these type 2 MAGEs, among them MAGE-L2, that are highly enriched in the brain. And MAGE-L2 in particular is highly expressed particularly in the hypothalamus, in human and in the mouse. And the gene resides in chromosome 15, human chromosome 15, in particularly in this Prader-Willi syndrome region of chromosome 15 Q11 to Q13. This region encompasses 6 coding genes and 6 non-coding RNAs that are deleted in a block. Different deletions lead to this development of Prader-Willi syndrome. You can see also Macaron 3 here as well. It will be mentioned in the later talk as well. And what is interesting for this region, that is maternally imprinted, and genes have to be expressed from paternal chromosome, as shown here on the left side. So healthy individual will have undisturbed chromosome 15 from the father. If this region is deleted, or even some genes mutated, this can lead to different neurodevelopmental syndromes, including Prader-Willi. And what is Prader-Willi syndrome? That's a rare genetic disease. It's one of the most common rare diseases, and it affects like 15,000 to 20,000 individuals, 1 in 15,000 individuals, and it presents in 2 different phases. So these children are born very hypotonic. They don't thrive well, they don't eat well, but then, and they are also smaller, they have specific facial features as well, but then after a couple of years, this switches and they become hyperphagic, and that's the major problem of these patients and the families, because they have completely uncontrolled hunger, leading to morbid obesity, and if it's not controlled, and all associated metabolic diseases. In addition to that, these children have also several other hormonal disbalances. They are they have small gonads, they have short stature, so several of them suggest dysfunction of neuroendocrine system. So, as I mentioned, MACHL2 is also in this region of these Prader-Willi syndrome genes, and interestingly, when we started to study this gene like a decade now or even more now ago, several patients were discovered that have mutation, point mutation, truncated mutation in this gene, and these mutations also lead to similar syndrome as Prader-Willi. Now it's called Schaff-Young syndrome, and both syndromes share several phenotypes, including neonatal hypotonia, and then later on, hunger control, growth hormone deficiency, intellectual disability, developmental delay. So several of these hypothalamic or neuroendocrine dysfunctions are presented in Schaff-Young syndrome patients as well. And in addition, also mouse and rat models were developed that also replicate several of the symptoms, suggesting that hypothalamic dysfunction and hormonal imbalance, it's one of the driving symptoms in this pathogenesis. So we are using this mouse model to address this question. And so when this gene was discovered, or because this gene is in this region, right, was already attracted a lot of attention, that we wanted to understand what this protein is doing on molecular level and cellular level. And I'm summarizing several discoveries here from my previous lab, as well as some other labs, where it was shown, we showed that MACHL2, this protein, binds to E3 ubiquitin ligase TRIM27 and deubiquitinating enzyme USP7, and together they regulate ubiquitination of protein WASH that regulates this retromer. And this seems like very complicated, and just to make it short, I want to let you know that what this does, it's controlling recycling of proteins, in particularly plasma membrane proteins and some secretory proteins as well. So, and if MACHL2 is not present in the positive cells, this recycling is disturbed, and cells, proteins end up in lysosome and are degraded. And of course, next question was which proteins require MACHL2 for proper localization and function in the cell. And we found several membrane proteins, including integrins, mannose 6-phosphate receptor, as shown here, using knockout approach, and then followed by immunofluorescence and flow cytometry. And in this cancer line, this led also to decreased viability of cells. However, the next question was physiological relevance, right? Normally, this protein is in hypothalamus. To address this question, we wanted to see which proteins are depleted in the hypothalamus of these mice, and surprisingly what we found is that not necessarily so many transmembrane proteins, but we found several secreted hormones and neuropeptides, and interestingly, several proteins that are core components of secretory granules. And what are secretory granules? So what I'm showing you here is sort of a schematic representation of secretion process in professional secretory cells like neuroendocrine cells in the hypothalamus. So you know every cell has these constitutive secretion pathways where some proteins are all the time secreted, but then this on the top is this regulated secretory pathway where neuroendocrine, like hormones, neuropeptides are accumulated in these secretory granules, are condensed, and they wait for the signal to be secreted. And several proteins are important for that this granule matures and is able to condense these proteins to this level and then also respond to the signal, including several granins, and also during this process, prohormones are matured, and hormones and peptides are developed or prepared by several endonucleases like PC1, 2, 3, and PC2. And chromogranins are proteins that are critical for condensation and also sorting of these proteins. And interestingly, these were the proteins that we found depleted in knockout animals, what led to decreased neuropeptide and hormone levels, and also decreased number of secretory granules in hypothalamus and different cellular models from the patients, suggesting that MHL2 is required for the regulated secretion of hormones and neuropeptides in the hypothalamus. And so this discovery somehow suggests that MHL2 is a tissue-specific regulator of retromer-dependent protein recycling and hypothalamic neuroendocrine function, as this is a heat map where these different genes, including MHL2, are expressed. And interestingly, MHL2 is expressed in the neuroendocrine tissues where these proteins required for these regulated secretions are present, whereas other parts of this secretory pathway are, other proteins or genes are expressed more broadly. And when MHL2 is not present in the hypothalamus, recycling of these proteins is abrogated and leading to decreased hormonal level. So this led us to the hypothesis that MHL2 somehow evolved in mammals for helping with better adaptation to diverse alternations from the environment. I didn't tell you for some other phenotypes, including that these mice and patients have also disturbed circadian rhythm. They have also impaired adaptation and anxiety to novelty in environment. So all of this suggesting that adaptation to ever-changing environment is perturbed. And in our future work, we are trying to figure out how is this happening and how we can overcome the requirement of MHL2 to try to find therapeutic opportunities for the patients. So with this, I would like to acknowledge, so this work was done when I was, a lot of them when I was still part of Dr. Ryan Potts's lab, now in Amgen. Now I'm at Texas Tech. I have collaborators here at the University of Ljubljana and also several universities in the United States and funding. And of course, nothing is possible with a great group. So these are my students here from my lab. And thank you for attention, and I'm open for the discussion.